everyone, myself Shetakar, and today we are talking about vision. So there's some learning objective of this presentation is first is functional anatomy of eyes, image forming mechanism and defects, structure of photoreceptor, mechanism of phototransduction and visual pathway, and finally the visual cortex. First we talking about the functional anatomy of eye. The eye are complex sense organ capable of receiving visual images which are then carried to the brain. So the some components of eye we see all this component in this diagram. In the posterior part of the eye, the outermost layer is sclera, which is provide protection to the eye, and the sclera is continuous in the anterior side of the eyeball. This is known as this is the transparent layer. This is known as cornea. And inside the sclera there is choroid layer is present, which is rich in blood vessels and provide nourishment to the eyeball and eye. And inside the choroid layer, the innermost layer is retina, which is contain photoreceptor rods and cones, and inside and on the retina there is a small depression. This is called fovea centralis, which is it contains large amount of cone cells, and this is optic nerve which is carried the visual sensation to the visual cortex. And the anterior portion of the eye, all are structure present. In the cornea there is a liquid film. This is known as aqueous humorous, and inside the cornea there is a iris. Iris is present and it can continue with the choroid layer of the eye. And in the iris there is a central aperture. This is known as pupil, which can provide the light into the eye. And posterior to the iris there is a lens is present, which is elastic, and the lens are hold with these ligaments. This is known as suspensory ligament, which are continued to the and the suspensory ligaments are attached on the ciliary body. Ciliary body contains ciliary muscle and ciliary process. Ciliary process synthesize aqueous humorous. And posterior part, so this is all the posterior part is filled with the vitreous humorous. So this is all about the structure of eye. Now the image forming mechanism. The image of the object is focused on the retina by following mechanisms. First, refraction of light rays by corneal lens. Second, ciliary muscle activity causing accumulation of lens. And third is change in pupil size by iris muscles. So the formation of an image on the retina. The eye is inverted and reversed with respect to the object. However, the mind perceives object in upright position, dip side and upside down orientation on the retina, because the brain is trained to consider an inverted image as normal. As we see in this first diagram, the object is normal, is in this normal condition. The light rays go through the lens and then the image focus on the retina is inverted, and the signals from the optic nerve go to the visual cortex of the brain, and picture is seen as normal, same as the object. And the eye is similar to the camera. Eye and camera are similar circuit of similar type of the similar to the camera. What happened in the camera, the object is present and the light rays go to the inside the camera, and inside the camera the image is inverted. As we see, the image is inverted inside the camera, and signal capable in the visual monitor the image is seen as normal, as real as object. Now the first is refraction of light rays. Refraction is the bending of light rays as they pass through one medium to another. Example, air to water. The cornea and lens bend, refract light rays to focus them on the retina. So the degree and direction depends on the angle at which the rays strikes the surface of the second medium. Difference in density between two medium. When the rays pass from a less dense to dense medium, example, air to water, bending is toward the midline, and it depends on the length of light. Light rays of longer wavelength get refracted more than light of shorter wavelength. So this is the diagram of the refraction of light. In the A diagram, the parallel uh, light rays come to the convex lens, and they can be focused on a particular focal point, and the light can be refracted. The bending of light and the rays are focused on one point, and in the convex lens, the parallel light rays are meeting at the point it can converge from the biconcave lens, and there is a bending of the light rays, and there is a bending of refraction of the light rays. Now the accommodation. Accommodation is the mechanism by which the eye changes refractive power by altering the shape of lens in order to focus objects at variable distance. Near point, nearest point of eye at which an object can clearly seen. So now that we see what happened in the accommodated eye and normal eye. So in the left side, this is normal eye and this is accommodated eye. In the first we see what happened in the normal. First we see the normal eye. In the normal eye or the relaxed eye, ciliary muscles are relaxed and jonular fibers or suspensory ligaments are under tension. So the lens is flattened and the lens is flattened, and in the accommodated eye, ciliary muscle contract and suspensory ligaments are relaxed and the lens is bulged and shorten of the lens. It can help to focus the near object on the retina because of the bulging of the lens. And bulging of the lens can increase the refractive power of the lens, so the light rays focus on the retina by the near object. So the nearest point of the eye at which an object can be clearly seen. Near response when changes focus from distance to near object. 3 changes occur in eye. Increase in curvature of the anterior surface of lens. As we see, the curvature is increase in the anterior surface of the lens. Convergence of the visual axis as the eyeball rotates medially more near the object greater the inverted rotation. So in the normal eye, the eyeball is parallel or in the center, but in the accommodated eye, the eyeballs are move medially to the eye. Constriction of the pupil are constricted in the accommodated eye, and in the normal eye pupil will be dilated. Change in pupil size. In bright light, pupil constrict. It is known as meiosis to a minimum diameter of 2 mm. In dark light, pupil dilate mydriasis to about 8 mm. The optimum vision is achieved with pupillary diameter of 2 to 3 mm. So in the as we see there is an iris, and inside the iris is the black this is the outer area is iris, and in the center of the iris there is a pupil. The black area is the pupil, Iris has two type of muscle, dilator muscle and sphincter muscle. So what happened in the constriction, pupillary constriction? The sphincter muscles are contracted and dilated muscles are relaxed in the constriction of pupil, and in the dilated pupil, dilatory muscles are contract and sphincter muscles are relaxed. So in the bright light, constriction of pupil, and in the dark light, dilation of the pupil. Defects of image forming mechanism. First is myopia, nearsightedness. First is myopia, nearsightedness. Far away object not visible clearly because parallel light rays from distant object are focused in front of the retina because of elongated eyeball, and it is corrected by using biconcave lens. So as you see, this is a normal eye. The parallel rays goes from the lens and it can focus only on retina. But in the myopia, what happens? The parallel rays goes from the lens and it can focus in front of retina because of elongated eyeball. And when we use biconcave lens, the light rays are diverted. The light rays are have bended. Light rays are bended and it can focus only on retinas. So the second is hypermetropia, farsightedness. Near object appear blurry because light rays are focused behind the retina because of shortened eyeball, corrected by using biconcave lens. So this is hyper. So in the D diagram we see the hypermetropia eye. In hypermetropia, the light rays are goes and the focus on behind the retina. We can correct it by using biconcave lens, so the light rays focus only on retina. Third is astigmatism. Astigmatism rays are focused on either in front or behind the retina. Objects appear blurry, far away or nearby. If lens curvature is not uniform, cause blurred vision due to continuous rubbing of eye, corrected by using cylindrical lens. Presbyopia, eyesight of old age, in which distant object clearly visible but nearer objects appear blurry, similar to hypermetropia. So in the hypermetropia, same thing happen. Light gets things happen. Light gets focused behind the retina. In hypermetropia, due to shortened eyeball, but in presbyopia because of age, loss of plasticity of the lens, corrected by using convex lens. Now the structure of photoreceptor. Retina contains photoreceptor cells, rods and cones. Each rod and cone is divided into an outer segment, inner segment with nuclear region and synaptic zone. So the A diagram is rod cell and B is for cone cell. This is outer segment, this is inner segment, and the nucleus present in between the inner segment and the lastly the synaptic terminal. In the inner segment, mitochondria is present, and the inside the rods the discs are present and cones are sacs. So the rods are activated in dim light, and this is responsible for scotopic vision, and these are help in scotopic vision or night vision. Long, slender and cylindrical shape, broad discs are closed, presented in periphery of eye, slow recovery time, cones, bright light and phototopic vision. They are activated in bright light and responsible for phototopic vision or the day vision. Conical appearance, cones discs are partially open present mostly in fovea. Fast recovery time, 3 types cone cell, red, green, blue cones. Photopigments bearing region is outer segment. So that what is now the photopigments. Rod pigment is rhodopsin. This is also known. It is also known as visual purple. It consists of two molecules joined together. They are retinine, aldehyde of vitamin A, and opsin. It is G protein couple receptor for light. When light falls on rhodopsin, a series of chemical reactions break the molecule rhodopsin into retinine and opsin. So the second is cone pigments, photopsin. 3 type of cone pigment differ in wavelength of light. Red sensitive pigment respond to long wavelength of light 560 nanometer. Green sensitive pigment responds to middle wavelength of light 530 nanometer. Blue sensitive pigment respond to short wavelength of light 420 nanometer. Melanopsin found within the ganglion cell layer in the retina plays an important role in non-image forming visual function including hormone secretion, etc. Mechanism of phototransduction. Phototransduction, the convergence of light energy to neural signals by the visual pigments in the photoreceptor. Phototransduction in dark and phototransduction in light. So first is steps of phototransduction in light. In dark, photopsin present in form of 11 cis retinine resting membrane potential of photoreceptor is minus 40 millivolt due to dark current. CGMP dependent sodium channel in outer segment is open due to sodium enters cell, decrease resting membrane potential. Inner segment sodium ion continuously pump out by sodium potassium ATPase maintain intracellular sodium concentration. Thus there is continuous flow of sodium ions from the cell interior to the cell exterior and from here there again into the cell interior. This is known as dark current. As we see in this diagram A, in the dark the CGMP dependent sodium channels are open. So the sodium are enter inside the cell, inside the outer segment of the photoreceptor, and the sodium can go higher concentration to lower, means outer to inner, and the sodium and in the inner segment the sodium potassium ATPase pumps are continuously pump out sodium, and these sodium from the outer environment of outside the cell, it's go again inside into the outer segment to the CGMP sodium channel, and this cycle is continuously flow. So this cycle is continue in the dark, so it is known as dark current. Steps of phototransduction in light. When light is put on the photoreceptor rods and cones, or in the rods, isomerization of 11 cis retinal to all transretinal, conformational change in opsin forming metarhodopsin. Second, activation of G proteins, transduction, activation of and then activation of diesterase that converts CGMP to 5-GMP and can decrease cytoplasmic level of CGMP, leading to closure of CGMP gated sodium channels. Decreased sodium influx causing hyperpolarization, decreased release of synaptic transmitters, response in bipolar cells. In the dark, there is increase the release of synaptic or glutamate inhibitory synaptic. So in the light, the sodium channels are closed in the outer segment. the CGMP dependent sodium channels are closed and the sodium ATPase pump is continuously pump out the sodium, so the cell will go in the hyperpolarizing state and the neurotransmitter decrease the release of neurotransmitter by the synaptic terminal. Changes occurring in retina during light and dark regeneration cycle of photopigments. When lights comes on the rhodopsin, 11 retinal can isomerize in all trans retinal in metarodopsin, and then separation from opsin this is known as bleaching, because the separation of opsin can release of reddish light or reddish, and it can convert in pale yellow pigment. All transretinal convert in 11-cis retinal by retinal isomerase in pigmented epithelium in retinal isomerase. Then 11-cis retinal in pigmented epithelium combines with opsin in the outer segment, and the rhodopsin is, and again the 11-cis rhodopsin is formed. Now the visual pathway. Visual receptor in retina. The visual signals from visual receptor in retina goes to the bipolar cell, and from the bipolar cell to the ganglionic cell, and the signals from ganglionic cell to optic nerve and to the optic chiasma, and then optic tract and the lateral geniculate body and optic radiation, and to the optic radiation, and lastly to the visual cortex. Now the diagrammatic representation of visual pathway. In the left side of diagram we see there is a left eye and right eye, and the visual signals from ganglion cell to go the optic nerve and optic chiasma, optic tract, and lastly lateral geniculate body and visual cortex. And we see the effect of lesion at various level of visual pathway. So first, lesion of right optic nerve. So the first lesion is right optic lesion in right optic nerve. So the when the lesion in right optic nerve, the right visual field is loss of right visual field, because the fibers on the right optic nerve come from the nasal and temporal field of right eye, and the lesion of right visual field. Lesion of optic chiasma produced by lesion of optic chiasma produced by temporal hemianopia. So now lesion of optic chiasma, what happened in that condition? The fiber from the both fiber of temporal field of both eye can so the loss of temporal field of the both eye, and as we see in this B diagram, the loss of both temporal field, loss of vision in both temporal field, and lesion of right optic tract. So lesion in right optic tract, into the right optic tract, the fibers are come from the right nasal field and right temporal field and left temporal field. So the right nasal field and left temporal field fibers are and lesion on these fibers. So the so the loss of vision of left temporal field and right nasal field and lesion of right geniculocalcaline tract. It is similar to the left homonymous hemianopia. So the picture, so these are some pictures of lesion of visual pathway. In the normal eye, we see clearly through the both eye, and in bitemporal hemianopia, so that both of eye temporal field vision is lost, and left homonymous hemianopia, left homonymous hemianopia, in left homonymous hemianopia, what happened? The temporal field of the left eye and nasal field of right eye are filled. The vision of temporal field of left eye and nasal field of right eye are lost. So the person can see like this. Now the processing of visual signal. Response of the retinal neurons, photoreceptors, hyperpolarizing response. Response of retinal neurons. Photoreceptor. In the photoreceptor, hyperpolarizing responses generate. It can go to the bipolar cell, and bipolar cell have depolarizing and hyperpolarizing potential. It can go to the horizontal cell. Hyperpolarizing membrane potential are generated on horizontal cell, and then can go amacrine cell. It has depolarizing response. Go to the ganglion cell. Ganglion cell produce action potential propagated along axon of optic nerve. So the direction of visual processing. So the direction of light is goes from the lens to the retina, and in the retina, so these are the layers of retina. As we zoom in this part of retina, the light is first go through the ganglion cell, then bipolar cell, and then photoreceptors. But the visual signals are processed. So but the direction of visual processing is from photoreceptor to the horizontal cells. They are interconnecting cell of the photoreceptor, and to the bipolar cell, and then amacrine cell. They are interconnected cells of bipolar cell. The amacrine cell, they are interconnected cells of ganglion cell, and then impulses come through the ganglion cell. Their action potential is generated. It is goes from the optic nerve to visual cortex. Now it's lateral geniculate body. Most of the fiber of optic tract terminate in lateral geniculate body of thalamus. Lateral geniculate body divide into six well-defined layers numbered 1 to 6 from ventral to dorsal direction. Input from nasal retina of contralateral eye projects to layer 1, 4 and 6. Input from temporal retina of ipsilateral eye project to layer 2, 3 and 5. The fibers in layer 3, 6 fibers in layer 3, 6 carry the impulses regarding color, texture and shape and finer detail. So So the broad line, so the dotted line is represent the ipsilateral fibers to the 2, 3 and 5, the layer of 2, 3 and 5 of lateral geniculate body, and the broad line is, the fiber of broad line is contralateral, the input from the contralateral nasal retina eye project to the 1, 4 and 6. The fiber from, the fiber from left geniculate body run to visual cortex, which consists of magnocellular and parvocellular pathway. Magnocellular pathway projecting from layer 1 and 2 of lateral geniculate body, and parvocellular pathway projecting from layer 3 to 6 of lateral geniculate body. As we see in this diagram, the fiber from 1 and 2 layer of lateral geniculate bodies are go to the visual cortex is magnocellular, and 3 to 6 are parvocellular cellular. So this is the organization. So these so in this flowchart, the ganglion cell are two type, M and P ganglion cell. The M ganglion cell project to the magnocellular lamina of lateral geniculate body, and the project and the magnos, and then projecting to the superficial layer of visual cortex. Their function is movement, location, space, organization. And the P ganglion cell can project to lateral geniculate, project to parvocellular lamina of lateral geniculate, and it can project to deep layer of visual cortex, and the function is shape and color. And from the parvocellular laminae, the interlaminar cells are arise by dendrites, and they can project into the blobs in the blobs of visual cortex and responsible for, and the function is color. Blobs are cluster of cells in visual cortex receiving information regarding color of an object. So now the visual cortex. Primary visual cortex V1 is the Bradman's area 17, located on the sides of the calcarine fissure. It is also known as the visual sensory area, as it receives the sensory information regarding vision. Macula has a much larger cortical representation than peripheral retina. So in this diagram, there is the upper peripheral quadrant of retina, upper quadrant of macula, lower quadrant of macula, lower peripheral quadrant of retina. So the area of macular quadrant is larger than peripheral retina. Where the fovea central is present in the center of macula, which is the small area in the posterior part of the eye, and their cortical representation is larger, because the fovea central is contain cone cell, and they are responsible for color vision. Primary visual cortex. Visual cortex divided into 6 parallel layer number 1 to 6. And these have 3 type of cells: simple cell, complex cell, hypercomplex cell. Simple cell respond to points of lights or bars of light in a particular orientation. Complex cell respond to bars of light in a particular orientation moving in a specific direction. Hypercomplex cell respond to bars of light in a particular orientation moving in a specific direction and of a specific line length. So these all are the 6 layers of the primary visual cortex in this diagram: V7, V3A, V3, V2 and V1. All these are the layers of visual cortex. So the function of visual projection area in the human brain: V1. V1 is primary visual cortex receive input from lateral geniculate nuclei being processing in term of orientation, adjust, etc. V2, V3 and ventral parietal continue processing larger visual field. V3A is for motion. V4 detection of shape, color, texture of an object. MT medial temporal, or V5 processes direction of movement. V6 may detect direction and speed of bodily movement, and lateral occipital is recognition of shape. V7 representation of upper and lower quadrant of visual field, and V8 is color vision. Secondary visual cortex. Area 18 and 19, also called visual association areas, located on the lateral part of occipital cortex, receive information from primary visual cortex and are concerned with interpretation of the visual stimulus. They help in appreciating the finer attributes of the object, like projection of form, texture, shape, depth, location, and orientation of the object. Thank you.